We are now beginning the lectures that are going to go over the diversity of life. And the place that we have to start is going to be with microbes. They're going to be the oldest forms of life on the planet. And in this lecture, we will talk about how we think life began on the planet with the first organisms, and then go through a little bit of diversity with microbes. So the Earth is approximately 4.6 billion years old, and the first organisms on the planet are going to be what we call prokaryotes. It is believed that they evolved around 3.5 billion years ago, and there are two main groups of prokaryotes that we're going to talk about today. We're going to talk about bacteria, which is the group that we're going to spend the most time talking about, and they also are going to be the group that we see the most in the lab this week. But we also are going to talk about archaea, which is another group of prokaryotes. Now, early organisms on the planet evolved under conditions where there was no oxygen. So the very early Earth did not have oxygen, and it wasn't until about 2.7 billion years ago that oxygen production began. So oxygen is going to be a byproduct of organisms that are going to perform photosynthesis. So we've already discussed the process of photosynthesis, and one of the byproducts is going to be oxygen. And so as these organisms start to perform photosynthesis, they are going to be releasing oxygen gas into the environment. Now, many of the very early organisms evolved without oxygen present, and oxygen is quite a reactive element and basically doomed many of the very early prokaryotes because they were unable to survive with oxygen present. Oxygen, we've talked about it, how it is very reactive and wants to grab electrons, and unfortunately it can do damage to life forms. And so many organisms that evolved started to die out once oxygen started to kind of become a more dominant gas on the planet. Now, once the prokaryotes started to do photosynthesis, they also are going to have different types of metabolic modes. Okay, and so there are going to then be groups that are going to start doing cellular respiration that can use oxygen as part of the process. We think that the earliest organisms lived alone for about 1.7 billion years. And so during that period of time, you would have only seen kind of these very earliest organisms. And the group, the prokaryotes, which we're discussing, they are still here and they are in great abundance. And we'll actually talk about how they outnumber us by an enormous number. So this is going to be a video that you can watch. I'm not going to play it right here, but this will talk about kind of how oxygen on the planet changed how organisms lived. Now, the first eukaryotes are going to evolve after the prokaryotes because prokaryotes are going to be the earliest form. Eukaryotes are then going to evolve from prokaryotes. The earliest we can tell that you had eukaryotes on the planet was about 1.8 billion years ago. And these organisms are going to be composed of one or more cells, and they have to have a nuclei. So that's going to be one of the defining characteristics of a eukaryote. That's what the name eukaryote actually means. And so we're going to start having nuclei and organelles present in these eukaryotes. They evolved from prokaryotes. And the way that this is thought to have happened is that we had small prokaryotes, and then one prokaryote actually consumed another prokaryote and incorporated that prokaryote into its body, which ultimately kind of became reliant on the organism, and therefore we have kind of this structure inside of another of a cell, which is kind of the beginning of what we think of as eukaryote. So the first multicellular eukaryotes are thought to have evolved around 1.2 billion years ago. And it's thought that they were originally started with single-celled organisms that started to live in colonies, but then they became dependent on one another. Some of the modern descendants of these early colonial eukaryotes are seaweeds. Okay, and so they are going to be single-celled organisms that actually live in kind of a colony altogether. Now, one way to look at kind of the evolution of life on the planet is to take a road trip across the United States. And so we can start in Kamloops, which is up in Canada, and that would be around 4.6 billion years ago when the Earth was formed. And as we travel on our path, we first go south into Washington and Oregon, and it isn't until about 3.8 billion years ago that we have the oldest rocks formed. Okay, so those are the oldest rocks that we know that were formed at that point in time. Originally, when the Earth formed, it was molten and it was too hot to actually have rock formation. So it wasn't until the Earth started to cool slightly that we start to have rocks being formed. Once we travel down and hit San Francisco, we are now at about 3.5 billion years ago, and this is when the first prokaryotes are starting to be seen, and there are fossil record of that. If we travel down to San Diego and then head over east to Phoenix, at this point, we're starting to increase the level of O2 in the atmosphere, which is going to change the face of the planet and going to change the organisms that can live there. This is about 2.7 billion years ago. We're going to head further east to Oklahoma City. This is about 1.8 billion years ago, and this is when the first eukaryotes are thought to have inhabited the planet. And then we go kind of northeast to St. Louis, and we have the first multicellular fossils being found kind of 1.2 billion years ago. When we travel to the next location in Erie, this is when we start to have the first large complex multicellular organisms, and this is about 600 million years ago. We then are going to go through to Buffalo. This is when we think that plants are going to have colonized the planet. This is 500 million years ago. And then once we get to Albany, this is the mid-Mesozoic, about 180 million years ago, when we would have the time of the dinosaurs. And then once we get to Boston, this is approximately 0.195 million years ago. This is when we start to have the first humans. So this is when Homo sapiens would have evolved. If we travel back to the origins of the planet, for the first several hundred million years, the planet was extremely harsh, and there's not really any way that we think any type of organism could have survived on the planet. About 4 billion years ago, the Earth is still very violent, but there's starting to be some changes. Water vapor starts to condense and then condenses into oceans on the planet, which then allows for the cooling of the surface. There are still volcanic eruptions at this time, and they're belching these gases like carbon dioxide, methane, and ammonia, and those are going to start to be these inorganic molecules that are going to then be important for life at some point. So they're starting to be present on the planet. Now, at some point, life is going to begin to form on the planet, and there has been a lot of research done into how this could take place. Because if we go from no life to life, this is a huge step. And life is something that we would consider an emergent property that is going to arise from the specific arrangement of molecules. So how they interact will then be able to cause this kind of property that is more than the sum of all their parts. There's this idea of abiogenesis, which is a theory that states that life arose on Earth because of spontaneous natural means due to the conditions on the early planet. And so the early planets had these conditions that we no longer have today, but they would have been the perfect conditions for life to form spontaneously. And so life can come from non-living matter. We may never know exactly how the planet formed, but there's lots of research that's been done that has kind of figured out that there are possible ways to go from a non-living matter to living matter. One of the big things in this area of study is that biologists have to look into fields like chemistry, geology, and physics in order to kind of unravel the mysteries of the beginning of life on the planet. So here are going to be some of the evidence for very early life that's going to come from other fields like geology. So in this image that we have right here, let me draw it. So in this image, what we're seeing is a rock sediment, and we have these cone shapes that they've lined out, and those are called stromatolites. On the picture on the right, we also have what we call stromatolites, and what stromatolites are they are created by blue-green algae. And so they have these layers and layers of material that they deposit, and they basically fossilize. And so these stromatolites that are seen right here and right here and here are in rock that's 3.7 billion years old. And these rocks are found on Greenland. These are tiny little stromatolites that were only about the size of a quarter, and they believe that these are fossilized colonies of microbes. They would be some of the earliest fossils on Earth. Okay, so taking evidence from geology and trying to understand how this can actually preserve a signature of life. Now, one thing that we need to talk about is that there is this abiogenesis paradox, because one of the big findings in more recent times is that there is no such thing as spontaneous generation. And so we say that life comes from life. So if you have one cell, the cells are going to come from cells that were there before. And so you don't have just like the spontaneous generation. But from a very early time, from ancient Greeks to well into the 19th century, it was believed that life can come from non-living matter, and this is going to be called spontaneous generation. There's a really famous experiment by Louis Pasteur that showed that you can't actually have spontaneous generation. This involved kind of putting meat into a vessel, and if you left the vessel open to the air, at some point you'd see flies arise almost spontaneously from the meat. What they didn't understand was that when you have the meat open to the air, there can be very small little eggs for insects, and they will grow and eventually produce flies, but that's not spontaneous generation. Okay. They also believe that fish would evolve from ocean mud and other types of things like this. This image that I found has to do with, there was a recipe that was how you are able to produce, make flies and frogs out of nothing. You basically had to crush up some stuff, and if you put it in the right conditions, you would then have spontaneous generation of these different life forms. And so we don't believe that spontaneous generation today is possible, but it is believed that spontaneous generation could have happened on the very young planet. Okay, and so this was again the Louis Pasteur confirmed through experiments that all life arises from reproduction of pre-existing life. And so it is thought that spontaneous generation could have occurred on the very early planet when the conditions were very different. So today we have a high level of oxygen in our atmosphere, but oxygen is very corrosive and it will disrupt chemical bonds. And so on a very early planet, if oxygen would have been present, chemical bonds that are going to be essential for these first steps of life to occur would not have been able to happen. And so oxygen was absent from a very early planet, which probably was extremely important for the spontaneous generation of life. Energy sources are also going to be very different on the early planet. So we have obviously the sun energy, but during the very early life, there would have been lots of lightning, a lot of volcanic activity. Not just, we have volcanoes still, but they're they're randomly across the planet, there would have been volcanoes everywhere. And there would have been an enormously high amount of ultraviolet sunlight, okay? Which is, we still have ultraviolet sunlight, but we have less than we would have had early on when the atmosphere was different. So many biologists believe that it is very possible that early conditions on the planet would have been perfect for kind of the simple chemical and physical processes that would have created life. So this is going to be a video that will describe kind of early life on the planet and some of the conditions that would have needed to be present for life to spontaneously occur. So we're now going to go into the 4-stage hypothesis for the origin of life. Okay, and it's going to have these 4 steps that are going to kind of outline what needed to happen and in what order for us to get to the first kind of independent living unit that we would think of as a cell. So the first step is going to be the abiotic synthesis of small organic molecules. So we need to be able to initially make things like amino acids. We need to be able to make a nucleotide. Those need to be made, but they have to be made spontaneously. They cannot be made by kind of a living cell. So you have to be able to put kind of carbon, nitrogen, oxygen, hydrogen in some type of system that will then allow them to then connect in the ways that we have for amino acids and nucleotides on their own. The second step is that there has to be some way of joining these molecules. Okay, so we talked about monomers to polymers very early on in our semester. And so we're going to have enzymes that are going to facilitate that. But can this happen in the absence of enzymes with the right conditions? This third step, which is one of the big steps, is to then kind of have the pre-cell, which is going to be some type of droplet with kind of these phospholipids that are going to maintain an internal environment separate from the external environment. And the last is the origin of a self-replicating molecule. So this is going to be extremely important. We can have a cell, but if we don't have the ability to have self-replication, then we can't have this genetic inheritance that we see today. And so we have to see if that would be even possible. Now, all of these are speculative, but all of them have been tested in the laboratory. The first stage, which is the abiotic synthesis of organic monomers, is the step that has been studied the most. And you might be asking why I'm showing you these deep sea thermal vents, but we believe that this is actually one of the places that the abiotic synthesis of organic monomers actually began. One of the most important studies that's been done, and kind of one of the earliest, is this study by Stanley Miller in 1953. Now what he did is he built an apparatus that was able to mimic the conditions of the early planet. And he set this apparatus up, he let it run for a week, and at the end of that he found amino acids that had been created spontaneously. Now since that time when Miller did this, many other laboratories have also done similar experiments, and they have kind of used different atmospheric mixtures and have produced other organic compounds. So this is an apparatus that was used by Miller. So what you can see is that we have a sea of water in this part of the apparatus. The sea is heated, and there would have been kind of a high temperature at this early planet. And so you would have water vapor. The water vapor then travels up through this apparatus and goes to kind of the atmosphere of the apparatus, so on the planet. In the atmosphere, there is ammonia, there's methane and hydrogen gas, H2. There are sparks from electrodes that are mimicking kind of lightning. If you let that mixture then condense, and finally you can capture all of the materials made from this process in a kind of beaker, and you test it for chemical analysis, they found those monomers of the amino acids. And so every amino acid has been found can be produced in this way. And so this definitely is a possible explanation for kind of the very first step of life on the planet. So scientists have tested other hypotheses, and they want to know where on the planet could there have been kind of the perfect conditions for this type of scenario of making these organic molecules spontaneously. And so some of the locations are kind of submerged volcanoes, or deep sea hydrothermal vents, and some of them are that meteorites actually were going to contain the first organic molecules on the planet. So they would be on the meteorite, and then once they land on the planet, then they are going to have organic molecules. The deep sea vents are one of the big ideas, and in the video that was on one of the past slides, we'll explain about that. But what I wanted you to see in this image, and I'm sorry that there are watermarks, but they would have made me pay hundreds of dollars to use this image without it. But you can see in these bubbles we have the organic molecules. And so these organic molecules are the necessary first step. And we'll be moving on to our second step, which are going to then need to kind of harness what's made in these vents. So the second stage is abiogenesis of polymers. So if we have monomers, but we need to be able to start to make more complex molecules. And so the idea is that these small organic molecules were present on the planet, and then they are going to start to form polymers such as proteins or nucleic acids. Remember, in our bodies and on the planet, at this point, we are going to have enzymes and other cellular equipment that's going to help produce proteins and nucleic acids. But these very first abiogenesis of polymers has to happen without life. So abiotic means there's no life. So researchers have studied this and have brought about polymerization of monomers to form polymers, and they've done this spontaneously. What they do is they actually will have these monomers, and they then drip the solutions of the organic monomers on either hot sand, clay, or rock, which would have been present on the very early planet, and they are able to make what they call proteinoids. And so in this image, in the black and white, these are proteinoids, which are a polypeptide or mixture of polypeptides that were obtained by heating a mixture of amino acids. So there was no enzyme involved in the creation of these polymers. Now, the way that they think this works is that if you have these organic monomers, let's just say from those hydrothermal vents, they're in the water, and let's just say the water washes up onto a rock on the land, and heat will vaporize the water, and what you're left is concentrated monomers. Now, some of those monomers, because of chemical reactions, will spontaneously bond. Okay, and when they do that, they are starting to form polymers. It's believed that on the early planet, raindrops or waves would have kind of washed up those diluted organic monomers, and if they washed up on fresh lava or hot rocks, they could then link to each other, become and form polymers, and those polymers could then go back into the sea. The water, the ocean on the planet is going to be where life begins, and so kind of have this constant connection between the sea and life. If this happens at a great amount, then you could have these huge amounts of polymers that are floating around in the ocean, which will then lead us to our next step. The next major step is the formation of pre-cells. And so we know that the smallest unit of life that we say today is a cell. And so how do we get that actual cell? So this has to be a way for there to be isolation of a collection of abiotically created molecules within a membrane. So laboratories have actually done experiments that have created pre-cells, and they have done this and formed spontaneously with those abiotically produced organic compounds. Now, when they do this, the actual pre-cells behave like living cells. These are a couple of the properties that they will actually have. So these again are created kind of in a spontaneous way, but those pre-cells have selective permeable surfaces, which our cells have selectively permeable membranes. They grow when they absorb molecules, and so we've seen that cells that are absorbing water or anything can get bigger. And they also will swell or shrink when placed in solutions of different salt concentrations. So they behave like we see the cells for isotonic, hypotonic, and isotonic. Over here in this image, what we have are these green fluorescent balls. Those are actually the prebiotically synthesized heterogeneous polyester microdroplets. Okay, and so what those are kind of these primitive compartments that we would call pre-cells. So it is possible to create these in an abiotic system. So one of the most important kind of defining characteristics of life is that there is this process of inheritance. And we have the self-replicating molecules. So we're able to have DNA that can replicate itself. We can transcribe our DNA into RNA and make proteins. But how do we get to that point? Because that's a very complicated system. How did we go from not having replication to then having self-replication? And so this is kind of one of the big stages and one of the most difficult stages to kind of understand. Now, it's thought that this can happen just in kind of slow, gradual steps. And the molecule that is believed to be the first kind of inherited or self-replicating molecule is RNA. So RNA can actually replicate itself without the assistance of proteins or enzymes. The RNA itself can act as its own enzyme, basically, and they're called ribozymes, so it would be these sequences of RNA. And so RNA itself can help other RNA replicate. And so these ribozymes are thought to be kind of one of the important steps in this process of self-replication. So in a laboratory setting, you can actually put RNA monomers in the right conditions and they will start to kind of self-assemble into these short sequences of RNA. And we can kind of think of them as genes. Now, you're going to then have a population of RNA and each of them is going to have a random sequence. Okay, so they're going to have, on this image we have kind of 4 different sequences of RNA. Now, some of these molecules will be able to self-replicate, and their success will vary. Some of them are going to be able to self-replicate very well, other ones won't. Those that can will then be the ones that replicate fastest, and they will increase in frequency in the population. Okay, and so again, this is kind of one of the most complex steps of how this process of life arose. But the main thing to know is that we think RNA is going to be kind of the original genetic material. Now, through natural selection, those very early pre-cells that contain some of that self-replicating RNA would have kind of actual genetic information that would help them reproduce more efficiently, and those would then increase in number. Okay, and so we can start to see how we can go from chemical evolution of just having these chemical compounds to then what we would consider Darwinian evolution. And so those pre-cells that were able to replicate themselves more efficiently and at a faster rate would be the ones that would then persist in the next generation. Now, this would have taken a very long period of time, and it's thought that kind of during the millions of years that we go with kind of these first very early steps of evolution of life, there then would have been the evolution of DNA, which is a similar molecule, but is more stable. Okay, and so over time, DNA would have replaced kind of the RNA, and the DNA would then be this kind of the set of instructions that we would keep separate and isolated, and it would store the genetic information, and RNA would then still be part of the system, but would not be the major genetic component. But RNA is extremely important in all actions of the cell, and so it's important to know that it's thought that RNA was the original genetic material. So now we're going to move on to kind of more about the actual prokaryotes and the ones that we have on the planet and kind of describe them. So we think the prokaryotes lived on the planet for about 2 billion years all by themselves. Okay, and during that time, they would have been kind of found wherever life is. Bacteria and prokaryotes can basically be found almost anywhere on the planet. They are going to have a collective biomass that is at least 10 times that of all eukaryotes today. And so if we kind of added up all of the prokaryotes and weighed them against all of their eukaryotes, they'd have at least 10 times more biomass. Prokaryotes can live in pretty insane conditions, places that it's too cold, too hot, too salty, too acidic, too alkaline for any eukaryote. And so again, you can find prokaryotes basically anywhere on the planet. Prokaryotes are also going to be one of the kind of big causes of human disease, and we'll talk about some of the different bacteria specifically that are going to be involved in human disease. One new area of study for prokaryotes is this idea of the microbiota, and this is going to be the community of organisms that is microorganisms that's going to live on and in and everywhere on the body. These organisms are absolutely essential for life, and it's believed that if we didn't have them, we would not be able to survive. And so some of the important functions that they provide for us are that they supply essential vitamins. They're going to allow us to extract nutrition from foods that we normally would not be able to extract nutrition from. They also will be able to decompose dead skin cells on our bodies. And they are going to be the first line of defense against other microorganisms that are actually intruders. So our body is completely covered in these kind of guards, these microbiome that is covering our body and protecting us from getting infected with these other intruders. And the prokaryotes are also going to play an enormously important role in the environment. They are going to decompose dead organisms and other waste materials. They also are going to help chemical elements cycle throughout the environment. Without prokaryotes, we basically would have dead, everything that died on the planet would just sit there and not decompose. Okay, and so prokaryotes are going to allow for that process to happen and basically are recycling these kind of organic molecules into the form that we can then use again. As I mentioned before, prokaryotes can live in places that no other organism can live. So for example, we have this thermal pool that is in Yellowstone. It's about 456 degrees Fahrenheit and it's extremely acidic. If a human falls into one, and apparently this happens quite often because people don't follow the rules, it will disintegrate the body very quickly. So no eukaryote can live here, but there are some prokaryotes that will actually live in these thermal pools. Now, one thing when we compare eukaryotes and prokaryotes is that prokaryotes are going to be extremely small. There are sometimes going to be ones that are a little bit larger, but on average, they're going to be much smaller than your typical unicellular eukaryote. And so here is the tip of a pin magnified to 605 times, and you can see the little tiny yellow specks are going to be the prokaryotes. Most likely these are going to be bacteria. Now, for the structure of prokaryotes, we've already talked a little bit about this, so it's important to just remember those main things, that prokaryotes are not going to have membrane-bound organelles. They're not going to have a nucleus. They do have a plasma membrane, and they usually are going to have a cell wall that is around the plasma membrane. Now, one thing about this group, the prokaryotes, is that they're extremely diverse, and you can find lots of different kind of forms. They can look very differently. About half of them will have some type of flagellum, which are these tail-like structures. They look very similar to what we would see kind of on a sperm cell, but these are the flagella. They are going to allow for bacteria to be mobile and kind of move around. Now, these are going to be the three types of prokaryote form that you need to know. These are all bacterials, and this is going to help you with lab material as well. So we have these three different common types, and you're going to need to know their real name. So this first type is called spherical, but the real name that you're going to have to know is going to be cocci. So we have these circles. The next type is a rod-shaped, which is called bacilli. And again, it looks like a rod, it kind of looks like a hot dog. It's called bacilla. So make sure that when you're on a lab practical that you say bacilli. The last type, which sometimes are called spirillum, but the book calls them just spiral. This is going to either be curved or spiral. So it looks kind of like sometimes like a corkscrew, but that will be your spiral type. Okay, and so all bacteria are going to fall into one of these categories. Now, most prokaryotes are going to be unicellular. That is kind of a defining characteristic. They are also very small, but you will sometimes find prokaryotes that will form what we call true colonies. And that means that you have bacteria living together, and there are sometimes specialized cells. And so, for example, if you look at this cyanobacteria, there are these cells that are slightly different, and each of these is an individual cell. They're unicellular, but they're living together, and so some specialization is kind of showing this next step towards what we would then consider a multicellular organism, but they don't actually go that far. We also are going to have sometimes giant bacterium. So there are some bacteria that are very large, and so being small is kind of the average and the tendency, but you can sometimes have this giant bacteria. Now, one interesting things that prokaryotes do is that they will attach to surfaces, and they kind of form these very highly organized colonies, which we call biofilms. And these biofilms can have, one type of prokaryote or many types of prokaryotes. And so what we see in this image is actually multiple different types of prokaryotes. Now, biofilms can basically form on any surface, including your teeth. This image is actually of dental plaque, which is a biofilm. It is also a biofilm that will cause tooth decay. And so one of the reasons why you're always supposed to brush your teeth and floss your teeth is because if you let dental plaque sit on your teeth for too long, the bacteria will start to kind of eat away at your tooth. Biofilms are also going to be among one of the types of bacteria that can cause disease in humans. Here is a sponge that is highly magnified, and the colorized bacteria can be seen in the green, the red, yellow, and blue. And so there are lots of different bacteria on your household sponge, so that's why you want to make sure that you kind of don't use one for too long, because it will start to accumulate lots of bad bacteria. Now, the way the bacteria and other prokaryotes are going to reproduce is through a process called binary fission. This is basically just mitosis, but when we're talking about bacteria, we do call it binary fission. This can allow for very high rates of reproduction, and so some bacteria can basically reproduce exponentially. However, usually there's going to be some type of limiting agent in the environment that's going to prevent exponential growth. So usually what's going to happen is that there's not enough food or not enough space, and prokaryotes will also have waste products, and if there's too many prokaryotes living in one area and all their waste products are being dumped out into what they're living in, they will actually pollute their own environment. Now, one thing that can happen is if a prokaryote is in an environment that's really harsh and not amenable to reproduction at that time, they form this thing called an endospore, which is going to be a structure that has a thick coat around kind of the genetic material. And it will allow for this prokaryote to live for potentially hundreds of years in this kind of almost hibernation type of process. Okay. Now, unfortunately, endospores can be very dangerous, and we'll talk about them. So this slide is showing binary fission. You have a bacteria, it has a circular chromosome. The circular chromosome has to be replicated, and then we basically are going to have mitosis take place. I just want you to recognize the term binary fission as the mechanism for prokaryote reproduction. So a little bit more about endospores. So endospores, this kind of way the bacteria can survive in very, very harsh conditions. Well, those endospores can actually survive for very long periods of time, sometimes even centuries, and they can be very dangerous to humans if they come into contact with them. So some of the important ones are going to be tetanus, botulism, and anthrax. All of these can be deadly. Now, when we start talking about prokaryotic nutrition, there are many different ways to transform energy. And so there's a lot more diversity in how prokaryotes obtain their nutrition than you would see in the eukaryotes. So some species are actually going to be able to harvest energy from inorganic substances like ammonia and hydrogen sulfide. Okay, so things that can live in very harsh conditions, that ammonia and hydrogen sulfide are going to be dangerous to us, but they're going to actually be able to thrive and use that for energy. Other organisms are actually going to kind of have these relationships with other organisms called symbiotic partnerships. And these can happen with animals, plants, and funguses. We have lots of bacteria that live inside of our stomachs that are symbionts with us. So we kind of get mutual benefits from having them in us, and they get a benefit from being inside of us. So symbiosis is going to be this term that means living together, and it's the close association between two organisms. Now, sometimes these are kind of required, so the two organisms cannot live without each other, and other times they're just kind of, they're beneficial, but they can survive without them. So one example is an animal that inhabits these hydrothermal vents, and these are these giant tube worms, and they probably think they look like flowers or some other type of weird plant, but they are actually animals, and they are a worm. And these will live by those hydrothermal vents we talked about in the very beginning of this lecture. And the bacteria that live inside of the actual tube worms are going to be sulfur bacteria. So they're able to actually break down sulfuric compounds. So the animals will absorb the sulfur compounds, and then they will be able to break that down, and they can convert CO2 from the seawater into organic molecules. So they're providing some food for the hosts, and the hosts are providing shelter for the bacteria. Many prokaryotes are also going to be photosynthetic, and there are even some that can do something called nitrogen fixation. So some types of cyanobacteria, which are the photosynthetic types, they can do this process of nitrogen fixation, which is when they take atmospheric nitrogen and turn it into a form that plants can actually use. Now, some types of cyanobacteria that can do this will form symbiotic relationships with other plants. And so in this picture, we have an example of this symbiosis. We have a water fern, which is called azolia, which are these little floating plants on this water. And the azolia can actually live in areas with poor nitrogen because the cyanobacteria will allow them to have a nitrogen source. Now, one interesting thing about this plant is that for thousands of years, these tiny floating plants have been used to help boost rice yields. And the way that they're doing this is that these little plants are able to get nitrogen, and then they're also going to be able to kind of support the rice plants as well. Prokaryotes are also going to play a huge role in the environment. They are going to be these organisms that are going to allow us to survive. Without them, we would not be here. Okay, and so what they can do is that they will provide a lot of ecological services that are essential for us to be living. One of the things they do is that they are chemical recyclers. And so they're going to be able to cycle chemicals through the system and take them from organic forms when they're dead and then put them back into their organic components that can be then used again. They're going to be extremely important for the breakdown of all organic waste and dead organisms. Putting prokaryotes to work is going to be really important in kind of other ways that we deal with our environment. And so this term bioremediation is the use of organisms to remove pollutants from the water, the air, and the soil. And so as humans, we know that we are kind of big causers of pollution, and we've been able to figure out ways to have prokaryotes actually fix some of the problems that we have created. So, for example, the way that we have our water and treat our sewage water and kind of clean it is with the use of prokaryotes and funguses. And so when you go to water treatment plants, you can actually see the organisms that are going to be used to help clean the water. Another is to use prokaryotes to help clean up toxic chemicals. There are bacteria that can break down some really harsh chemicals, and there are even bacteria that can break down plastics, which is a really big area of study right now because we have a huge plastic problem in the world. There are plastic microparticles absolutely everywhere. We drink it, we're eating it. And so if we can start to figure out how to deal with that, I think prokaryotes are going to be one of the big tools. In this image with the airplane, this is actually an airplane that is spraying chemical dispersants on oil. And what this is going to be these prokaryotes that will actually be able to clean up and kind of break down oil into a non kind of damaging molecule. So we are now going to talk a little bit more about kind of the diversity of the prokaryotic branches, and then we will talk about kind of our next group, which is going to be our first eukaryotic group that we're going to talk about. So when you look at the prokaryotes on a molecular level, and so this is looking at the actual genomes, there are two major branches of the prokaryotic evolution. These are going to include bacteria and archaea. And so we have bacteria right here on the branch. This is going to be the oldest form of life. And then we have the second prokaryote, which is going to be called archaea. And that's going to be, I'll talk about them in a second. Now, the eukaryotes are going to be kind of the more complex group, which is going to include us. And so those are going to then evolve out of this bacterial group. We're actually a little bit closer related to archaea than we are to bacteria, although this is a very distant relationship. Now, we then are going to start talking about three domains of life. And so these three domains of life are going to be the highest classification that we will discuss in the diversity of life. Okay, these are going to be bacteria, archaea, and eukarya. So there's going to be the three groups of the three domains of life. They are not all prokaryotes because we now are including eukarya. And so when we talk about the three domains of life, every life form is going to fit into one of these three categories. Okay, we're mainly going to talk about eukarya because that's the group that we're found in. That's where we find animals, plants, and funguses. But we're going to mainly focus on the eukarya. Now we're going to talk a little bit about archaea. So archaea are really interesting because they're found in a lot of locations, but they also are found in some extreme environments. And so we'll talk about three kind of extreme places you can find archaea. The first is in extremely hot water. So there are a group called extreme thermophiles, which are heat lovers, that live in hot water like the thermal pool of Yellowstone. The next group is going to be extreme halophiles, and a halophile is a salt lover. And so these archaea can live in extremely salty conditions that most organisms can't survive in. And the third group are going to be these methanogens, which will live in areas that are going to have no oxygen. And they give off methane as a waste product. Now you can find these at the bottom of lakes, kind of in mud, but you also can find them inside the digestive tracts of animals. And so, for example, cows are going to have certain types of prokaryotes that live inside their gut that are then going to help them digest the cellulose of the grass they eat, but they give off large amounts of methane, and cows then burp all that methane out. Now we're going to talk a little bit more about bacteria, and specifically bacteria that cause disease. So any type of bacteria organism that causes disease is called a pathogen. And normally the way that bacteria are pathogenic is that they produce poison. Now there are going to be two different mechanisms of how they produce this poison. Some bacteria produce exotoxins, which are going to be poisonous proteins that are actually going to be secreted by the bacterial cells. Staphylococcus aureus will produce several toxins. Now, Staphylococcus aureus is found on our skin in our nasal passages, and it's normally there. But unfortunately, if it gets inside of a wound, it can actually cause a serious disease. And one of the problems is it can cause a flesh-eating disease when your skin will kind of just keep on sloughing off layers. There's another type of Staphylococcus aureus toxin that causes toxic shock syndrome. And you probably have heard of this one. Mostly it has to do with, everyone gets scared by using tampons. If you leave a tampon in too long, you can actually have Staphylococcus aureus toxin causes toxic shock syndrome. And if you get kind of a millionth of a gram in your food and you eat it, you can get diarrhea or vomiting. And so that's a very small amount. So even a small amount can cause quite a sickness. The other way the bacteria cause disease is by producing an endotoxin. And so the bacteria itself will have the endotoxin inside of them. They don't secrete them. So if you come into contact with the actual bacteria, then you will be kind of exposed to it. All of the endotoxin induce kind of the same general symptoms. They have fever, aches, and sometimes in dangerous situations you can have this very rapid drop of blood pressure, which is called septic shock. Now, septic shock can be triggered by an endotoxin, and one example of this is meningitis. So meningitis can kill a very healthy person in a matter of days or hours. And so it's a very dangerous bacteria. Another group that has endotoxins are salmonella. And you probably have heard of salmonella as being something like why you're not supposed to eat raw eggs, although it's very rare to get salmonella from eggs. Now, because bacteria have these endotoxin and exotoxins, they have been used as biological weapons for thousands of years. Now, anthrax is one of the most dangerous biological weapons, and the endospores can actually enter the lungs, and if they're in there, they can multiply and ultimately produce enough endotoxin that the individual will die from kind of accumulation of that in the blood. The plague, which was carried by fleas, the bacteria entered the bloodstream and then they would create kind of the toxins, and that would then kill the person. The pneumonic plague infected the lungs and was more dangerous because it would actually become airborne and you could have other people kind of breathe that in. The disintegrating lung tissue is coughed out in droplets. Very easily transmitted and very dangerous. This next one, Clostridium botulism. So this is an exotoxin botulism. Botulism blocks the transmission of nerve cells, and they will stop from muscle contraction, which basically will kill you right away because you have to have like your heart still pumping. So this is one of the deadliest poisons on Earth. 30 grams of pure toxin of botulism could kill every single person in the United States. So unfortunately, bioterrorism is something that has been around for a very long time. Many people didn't understand exactly how this was happening, but they were using what ended up being bioterrorism to kill other people. So during the Middle Ages, armies would take plague victims and throw them into the enemy ranks. Again, they don't understand how transmission of diseases are taking place, but they know that being around plague victims was able to cause more individuals to get the plague. When we had early conquerors and settlers and warring armies in the South and North America, they would give native peoples items that were purposely contaminated with infectious bacteria. Now, again, they didn't understand what bacteria were, but they knew that if they gave them items, they often would then have mass casualties. In a more recent bioterrorism, in 1984, there were members of a cult in Oregon that contaminated salad bars with salmonella bacteria to get a point across. And in fall of 2001, this is soon after 9/11, 5 Americans died from the disease anthrax, which was in a presumed terrorist attack. And now to something very different and hopefully more happy than bioterrorism, but we're going to start talking about kingdom Protista. And so this is going to be the first group of eukaryotes that we're going to talk about. So protists are going to be eukaryotes, and they're going to be the earliest eukaryotes that have evolved. They are thought to have evolved from prokaryotes about 2 billion years ago, and this fossil that we see that's about 55 to 34 million years ago shows a type of protist. Now, the very early primal eukaryotes were going to be the predecessors of a great variety of modern protists and all other forms of eukaryotes that are going to come from them. So protists are kind of the ancestral type, and then we have eukaryotes that are going to turn into plants, funguses, and animals. Now, the protists are a weird group because it's not really a normal taxonomic category where all of them have the same characteristics. So we're saying like all of these are in here because they have this, this, and this. They're basically all there because they don't belong anywhere else, and it's more of a catch-all group. So when they look at protist phylogeny, they keep on having new changes and new information which cause scientists to revise how they think the phylogeny of this group actually began. And so instead, it's more of this catch-all category. It includes all eukaryotes that are not going to be categorized as fungus, animal, or plant. And so anything that doesn't fall into those categories are going to fall into the protists. Most protists will be unicellular, but there are some examples of multicellular protists. Now, one mark of the protist group is that there's extreme diversity in almost every single facet of their lives, from kind of how they obtain their nutrition to where you find them to their anatomy. So there are going to be multiple different types of protists. We have photosynthetic protists. Some of these are going to be in this group that we call the algae. This will also include some cyanobacteria. So there are some cyanobacteria that are actual bacteria, and there are others that are going to be considered protists. Another group are going to be heterotrophs. So they're going to acquire their food from other organisms like us. So they're going to eat their food in that way. So some of them are going to be fungus-like, where they will actually absorb kind of molecules, and others are going to be parasitic. So some parasites are going to be like this trypanosome that we see kind of in this center picture. Let me just draw an arrow. So these are trypanosomes, and so they are going to be parasites. And a parasite is going to derive its nutrition from a living host, and it's going to harm that host in that interaction. There are also going to be other protists that can go back and forth, and they're called mixotrophs. So they can do photosynthesis when they need to, and they can be heterotrophs when they need to. So they're not kind of stuck in any one particular type of metabolic pathway. So protists will be able to live in a diverse number of habitats. The common theme is that they have to be moist. So many of the protists will live in actual water, in the ocean, lake, or pond, river, place like that. Others will be found on land, but they have to be somewhere where there's damp soil or leaf litter. And other types will be symbionts, which means that they're going to live inside the body of their host organism. And a symbiont means that it's going to be a positive relationship for both the host and for the protist. Now, because classification with protists is so messy, there aren't really kind of organized in a way that's telling us anything about phylogeny, but more kind of just how they have similar characteristics. So this first group we're going to call the protozoans, and this is going to be a group that are mainly going to be heterotrophs because they are going to ingest food, and they're going to thrive in lots of different environments, including aquatic environments. But most of them are going to eat bacteria or other protozoans, but some will actually absorb nutrients from the water. And these ones will also, some of them are going to be found as parasites in animals. Not a lot of them are going to be parasites, but they can, and some of them are some of the most dangerous in the world. So for example, this trypanosome is going to be an organism that can cause a pretty serious disease. So this first group of protozoans are going to be called flagellates because they have flagellum that are going to allow them to swim. Now most of these are going to be free living, which means they're non-parasitic. If you're parasitic, that means you are living inside of something and therefore not free. But there are going to be some parasites that are in this group. The top picture is of trypanosome, so not the red circles, those are going to be red blood cells, but the little kind of wavy looking structure, that is going to be the protozoan that will cause sleeping sickness. The bottom picture is going to be of Giardia, which is a common waterborne parasite that will give people kind of severe diarrhea. So you're not supposed to drink kind of unfiltered water, kind of like from a lake or stream, because you could get severe diarrhea. There are other flagellates that will live symbiotically with kind of other organisms. And there's a group that lives inside the actual digestive tracts of termites. So termites are really tiny, but there is going to be a protozoan that lives inside the termite that will help break down the cellulose of the wood that they eat into simpler molecules like glucose so that they can actually use them. Amoebas will also be found in the protozoans, and so they're going to be characterized by very great flexibility in their shape. They can have all different forms, and they don't have any permanent structure for locomotion. Most species will move by means of pseudopodia, which are those kind of fake feet, and they do this by kind of putting their cytoplasm into these, kind of pushing them into these pseudopodia and kind of moving their body with them. Others will form these things called forams, which are actually shells. And so the shell will then be on the outside of the protozoan amoeba, kind of giving it this interesting structure. This next group are going to be called apicomplexans, and they are going to be all parasites, and they can cause some serious human diseases. Plasmodium is one. This is a parasite that causes malaria, and so malaria is an absolutely horrible disease that is very dangerous in certain parts of the world. The next is toxoplasm, which is something that if you have a cat, you may actually come into contact with quite often. It has kind of a complex life cycle where it requires a feline host at one point. People will actually become infected with this if they handle the cat litter of outdoor cats. It doesn't seem to be a problem with indoor cats as much because they're not being exposed to this toxoplasm. But if you do come into contact with the litter of an outdoor cat, usually you won't become sick because your immune system will keep the parasite in check. However, there is one situation where it is dangerous, and that is when you have a woman that is pregnant. So if a woman is pregnant and she becomes newly infected with this toxoplasma, she potentially can pass that on to her unborn child, which then could actually suffer damage to the nervous system. Okay, because the unborn baby doesn't really have a very substantial nervous or immune system at that point, and so can have some more damage take place. The next group are called ciliates, and these are named because of the cilia, these kind of short hair-like projections that they use for locomotion, but also to sweep food kind of into what would be considered their mouth. Most of them are going to be free-living and live in water. One kind of famous example of the ciliates is going to be a paramecium, and you'll see that one in lab. And then there are going to be some that are heterotrophs and mixotrophs. So mostly are going to be heterotrophic, where they have to eat their food, but they can sometimes perform photosynthesis. The next group, which I am just fascinated with because I think they're so weird looking, are the slime molds. And slime molds are going to be these multicellular protists. They are related to amoebas, and they feed on dead plant material. They're going to be classified kind of based on their DNA. They originally were thought to be funguses. They do kind of look like a fungus, but they are not found in that group. They're actually going to be from a different lineage. There are two distinct types of slime molds. There is this plasmodial slime mold and a cellular slime mold. And I just wanted to mention the name of the two top slime molds. The first one is called a raspberry slime mold because it kind of looks like a raspberry. And the second one is called a pretzel slime mold because it does kind of resemble a pretzel in some ways. I can't remember what the yellow one was called. This plasmodial slime mold has a feeding body that you can see in the upper left-hand corner, this yellow kind of mass. And so it's an amoeboid mass, which kind of means it looks like an amoeba. And it will kind of have these pseudopodia that will then kind of extend out into leaf matter and decaying material on the forest floor. They will kind of enlarge and increase the surface area that they're extending, and they can come into contact with, food, water, and oxygen to help them. This bottom picture is actually the reproductive structure of the mold, and when it's going to go through the reproductive time, it's going to actually kind of build this other structure that will disperse spores. In cellular slime molds, the feeding stage is more of a solitary amoeboid cell, and so they don't kind of all clump together when they're feeding. But if food is in short supply, then they will kind of swarm together and form the slug-like colony that you can see in the center bottom, and kind of move together as a single unit. When the slime mold is ready to reproduce, they will then form a different structure that will then release spores. And you can see kind of these reproductive structures on the left and the right of the center picture. So here is kind of different stages of the amoeboid cells, where they are just kind of by themselves eating. If they become in contact with an environment that's not really kind of good for them, they will then form the slug-like colony, and then finally will produce this reproductive structure. This next group we're going to talk about are algae, and they can be found either unicellular or kind of in a colony. So algae are going to be protists and cyanobacteria who are going to do photosynthesis, and they're going to be extremely important for marine ecosystems and freshwater ecosystems. Right now, one of the big kind of pushes is to be able to have kind of algae start to convert light energy to chemical energy for us to produce biofuels. And so many labs around the world are working on kind of creating algae that can then produce biofuels for us to then use to power our cars and other machinery that needs that type of fuel. So there are many unicellular algae, and they're going to be kind of a component of phytoplankton, which are going to be mostly these photosynthetic microscopic organisms that will drift near the surface of ponds, lakes, and oceans. So they have to be exposed to the sunlight in order for them to be photosynthetic. There are going to be groups. One is called dinoflagellates. These are going to be species that have kind of this reinforced external plates made of cellulose that you can see in this first picture. They're then going to have diatoms. These have glassy celled walls that contain silica, which is a mineral they use to make glass. And so kind of they're made out of a similar component. And this last group, the green algae, which are going to be named for their green chloroplasts. So if you see them, they're green, they're going to be doing photosynthesis. And so volvox are going to be an example of this. I just wanted to show another image of diatoms and some dinoflagellates. I think diatoms are absolutely beautiful. These are just some examples of them. They kind of look like stained glass when you look at them under a microscope. This next group are the seaweeds, and they are going to be these large multicellular marine algae. They look very similar to plants, but that's only through convergent evolution. So they solved problems in a similar way. They are not closely related. They're actually closer related to unicellular algae. They're going to tend to grow on rocky shores, and there are going to be three main types that we have. It's mainly classified based on the pigments that are in their chloroplasts. And so we've only talked about one type of chloroplast, but there are other types that are going to have different types of pigments. So green algae is going to be this, the far left picture. We're then going to have red algae, which is in the center, and then brown algae, which is we're more familiar with, which is kelp. Kind of these long kind of strings that kind of look like leaves. Again, they are not closely related to plants, but they do look very, very similar to plants if you kind of compare them. Now, the last thing I wanted to talk about was kind of this evolution to multicellular life, because this is going to be kind of our next step. So we've talked about unicellular organisms. We've also talked about unicellular organisms that live together, but there's a kind of a big step that has to take place before we go from kind of colonial multicellular to true multicellular organisms. So an early multicellular organism has to have specialized cells. And those specialized cells can be something to do with feeding, for waste disposal, gas exchange, or protection. Okay, but what happens is that those two cell types have to be dependent on one another. So you cannot have kind of the different specialized cells living apart from each other. So they're specialized, but they are dependent on each other and will not survive on their own. There then is going to be kind of this next step, which would then have an organism which is going to start to have kind of gametes and somatic cells, and that would be what we would consider kind of a true multicellular organism. And so the earliest multicellular organisms are thought to have been colonial protists, and kind of this is going to be our next step into the kind of evolution of the diversity of life.